All right. Well, this is Dr. Jeannie Marazzo, the director of NIAID, and I want to welcome you all to the first of a series of online webinars that will highlight ongoing and planned activities for the new RECOVER Treating Long COVID or TLC clinical trials program. So the purpose of this webinar is to provide brief updates on how NIAID is proceeding in launching RECOVER-TLC, and most importantly, to provide an opportunity for the patient, patient advocate, healthcare provider, academic, and industry communities affected by long COVID to continue to share with NIAID your questions, comments, concerns, and ideas. This is a live webinar, but it's also being recorded and will be posted online so it can be viewed at later dates. The agenda for today's webinar includes introductory remarks by Dr. Julie Gerberding, CEO of the Foundation for the National Institutes of Health. She will be followed by a presentation by Dr. Joe Breen, NIAID's lead on long COVID and my delegate to the overall RECOVER program, who will describe recent NIAID RECOVER TLC activities since the September 23rd, 25th workshop that we and FNIH co-hosted. Joe's presentation will be followed by one given by Lizzie Gerling, Dr. Lizzie Gerling, Associate Project Manager, Translational Science at FNIH, who will provide an overview of the therapeutics and biologics that have been submitted to date through the new RECOVER TLC portal. Following Lizzie's presentation, we'll open the meeting for questions from participants. To maximize the number of questions that can be addressed during the webinar, we've accepted queue and comments ahead of time. Webinar participants also will be able to pose questions to presenters live during the webinar by entering them into the chat. We will address as many as we can as time permits. As we indicated at our September workshop, we will convene these webinars on a regular basis. Our next one will be held in January. We'll disseminate the date and time for that virtual webinar in the next few days so that you can add it to the calendar. I would like now to invite Dr. Julie Gerberding to provide comments on behalf of the Foundation for the National Institutes of Health.
Thank you, Jeannie, and thank you everyone for participating in this webinar. I can only tell you how hard the teams have been working behind the scenes since the summit in September to really respond to what we learned, what we heard, and what we think we need to do going forward. I can't really characterize all of the debates and discussions we've had about the best way to expedite progress in this sense, but I can say there are three main principles. One is that we have an open door. We welcome input and perspectives, particularly of people with lived experience, but also from the scientific community, the private sector, et cetera. The second thing is we have an open mind. We're coming into this without a lot of preconceived notions about what will work or what won't work in clinical trials. And we want to understand the evidence base that would lead us to proceed in a certain direction, but at the same time, have an open mind about what we don't know and acknowledge that we all have a lot to learn. The last thing I would just say personally, I think I speak for everyone, but we have an open heart. We understand how difficult this illness has been for so many people, and we care, and we are working really, really hard to move forward. One of the reasons that FNIH is involved in this process is because we did have experience working with NIH and many, many other stakeholders during active the process for adjudicating clinical trials for the acute COVID syndrome. And I think we're trying to learn the lessons that came into play in the aftermath of that so that we can improve our performance and our speed, but also be mindful of how valuable a collaborative approach really is. So thank you so much for joining the webinar, and I look forward to listening and learning as we go forward. And Joe, do I turn it over to you now?
Yep, thank you. Let me share my screen. Okay, can I ask a panelist, can you see my screen?
Looks good.
Great, thank you. So as Dr. Marazzo mentioned, my name is Joe Breen. I'm working at NIAID and helping to pull together this effort, working very closely with our FNIH colleagues, as well as others across NIH, our other partners, and soon to be those outside of NIH as well. So this is really to keep to the themes of what we heard at the workshop and trying to share and be as transparent as possible with where we are in the next round of clinical trials under the Treating Long COVID program. Let's see. So I don't think I have to spend much time on this slide, but this is a reminder how much we have yet to understand about long COVID and the many different scientific milieus needed to understand long COVID, which really indicates why we need an all hands on deck and lots of different experts helping as we craft this way forward. As Dr. Marazzo mentioned, we need caregivers, input from those affected by the disease, those taking care of them, as well as disciplines, from neurology to infectious disease and cardiology, because it really is remarkable, the tissue tropism of SARS-CoV-2, and unfortunately, the damage it's wrought, and we see much of this in long COVID. This RECOVER-TLC is part of the overall RECOVER network. And I just wanted to, oops, sorry about that. I just wanted to remind folks that, there are some guiding principles. Gosh, don't know why that keeps happening. That the key scientific aims of RECOVER are really to develop a national scale program to try and understand the long COVID illness overall, and really with the idea of understanding the pathobiology from observational cohorts, from autopsy studies, and from other tissue studies to try and tackle this and design the best intervention strategies possible. There are a number, 8 different trials underway, and those are continuing. Actually, some have completed enrollment, some even relatively recently, and we eagerly await the results, but we can't wait. We have to keep moving forward based on what we know today to initiate new trials, because as we all are aware, there are no approved therapies yet, unfortunately, for long COVID. This slide is to just represent that we had a number of experts from all across RECOVER, as well as actually some international colleagues at a meeting that Dr. Marazzo mentioned, and also Dr. Gerberding at the end of September, which was really brought together those aspects of what we knew of pathobiology, observational cohorts, electronic health records, which have actually contributed substantially to the understanding of long COVID, and then discussions thinking forward, like, for example, of the kind of endpoints that are needed. And really within days of the close of the meeting was the launch of a web portal where you're going to hear quite a bit more about what has come into that portal from Lizzie Geerling when I'm finished. So the goals of the workshop were really this high-level scientific review with an emphasis on community input at all stages to try and design forward and highlight the opportunities for trials based on what we do know. There are a number of trials ongoing, some with U.S. government support, some with private support. So we're taking into account that sort of landscape analysis, as well as the master protocols developed for the RECOVER 1.0 studies, and then thinking forward as to what the best opportunities are. And that is being the summary from that workshop, which is the workshop itself, of course, is publicly available, is being summarized and will be available for the public at a fairly detailed level, because we know it was a long workshop, particularly those that are are ill and might not be able to have attended the entire workshop. The topics were, as I mentioned, the pathobiology, the need for biomarkers, understanding epidemiology, and that has changed, of course, 3 years or 4 years after the onset of the pandemic. And we've also learned in the last year a lot more about what long COVID looks like in the pediatric and adolescent realm, and it does look different. There was a lot of discussion of clinical trial designs, intervention prioritization strategies, and endpoints. It was very well attended, which we greatly appreciate, because this will be, this is a public-private partnership between NIH, NIID, and FNIH, but will require a partnership with experts across NIH, across the US government, and the extramural and community, including patient groups and caregivers, really across the country. So what do the data suggest, and what do we hear at the meeting? We do keep hearing some consistent themes, and these are in the literature, in the projects that are funded in this area, certainly by the US government. And those are things about viral reactivation of herpes viruses like EBV, but also it's not always that clear a signal. I think we have to keep a little bit of an open mind yet, because it's not, we need to keep studying that. It's not an easy thing to do. Persistent viral infection, I have a few more slides about that. There have been consistent clues that lend us to believe that some people clearly do have at least viral antigens, and there's been, and I have a slide, there's some evidence for persisting double-stranded RNA from this virus as well. So I think we need to understand more, but what we do know more than we did even a year or two ago. And the other factor that goes hand in hand really is about immune dysregulation, which is commonly seen and can be measured, well described in acute COVID. And there's an interplay, I think, at play that with immune dysregulation. And I think that's where we're seeing a lot of commonalities to other infection-associated chronic diseases, such as ME/CFS, but not exclusively to ME/CFS. So I mentioned one of the findings that was talked about at length at the scientific meeting, and I think is still really being discussed and investigated in the community, is persisting viral antigens. This paper, which has been seen in several forms, here was accepted and is really a quite large study at this point, utilizing a RECOVER cohort as well as a cohort that existed prior to RECOVER, where they're led by a group at University of San Francisco, but also California, San Francisco, but also an assay group at Harvard led by David Walt and Zoe Swank, where they really have seen SARS-CoV-2 antigen detected in blood for up to 14 months. But they've also seen this in asymptomatic individuals. So I think what's fair to say is that they think that the blood, and it's in this paper, is potentially a way to measure what's going on, but may not be where this antigen actually is being shed from or where it exists. So it may be a way to dip in and understand what's happening in the pathobiology, but may not actually be the perfect way to measure it. But this group and others are detecting and using increasingly sensitive methodologies to try and understand this and correlate with clinical outcomes, which is still not that clear. This study was published earlier this year, which actually looked at tissue samples from patients in China, and they did see viral RNA that did persist in tissues for up to 4 months after infection. And they did have a correlation, that is an association with long COVID symptoms. So it's a smaller study. It needs to be replicated. But I think along with the antigen data that's been seen in several different studies, it lends us to keep looking hard, and we have to keep the hypothesis of potential viral reservoir or persistent infection. And there is also immunology data that would lend support to that. So I think these are the kind of things that we're tracking and taking into account, and we'll ask working groups to take into account as we decide whether we're going to look at an immunomodulator, for example, versus another antiviral, which, antivirals have different properties, of course. So that will also need to be factored in. Apologies for my clumsiness here. So what's really happened since the workshop? There's a report that's been generated. It will be published, and that will be available. The portal was available very shortly, within a few days at the close of the meeting. We continue to, with our partners, assess and monitor ongoing long COVID clinical trials, both in and out of U.S. government support, so that we understand the landscape. And we've established and are establishing a prioritization process and a triage and working groups to look at the interventions that have come in through the portal. And Lizzie Geerling is going to tell you a lot more about that in the next group of slides. We did also publish an RFI, a request for information, that was following the meeting, with a few different things in mind. To continue to remind people that we're going to take in these potential therapeutics. We wanted people to self-nominate to be on working groups that could help us prioritize within the kind of interventions that were proposed. We also are asking for suggestions for biomarkers that could potentially be validated, as well as general feedback on the process. Again, we're really trying to be transparent here and work with the community. And the working groups will contain, of course, as was mentioned at the workshop, members of patients' advocacy community, as well as caregivers, et cetera. So this, again, as Dr. Marazzo mentioned, is one of many periodic webinars we hope to share where we are. Of course, we expect this activity to ramp up as we initiate the prioritization and ultimately that we start the trials, and as well as a meeting to disseminate latest research and findings and really review overall progress, but really take input as well. Continue to take input and communicate with the community that is being so affected by long COVID. Thank you for your attention and to the teams that put this together to date, and I'll turn it over to Dr. Geerling.
Great, thank you, Joe. So I'm just going to jump right in so that we can get to the Q&A pretty quickly. But like they mentioned, I'm just going to kind of give a broad overview of some of the submissions that we've received to date, just generally speaking, who's been submitting, what kind of things are coming in, etc. So please advance. Okay, so I just want to remind everyone, oh, can you go back one slide? Thanks. Okay, so this portal is going to remain open for the foreseeable future. We don't have any plans to close it currently. And so basically that means that we're going to be reviewing agents on a rolling basis and what we're kind of referring to as waves. So we're hoping to perform the first wave of agent review before the end of this year, so likely mid-December. And as Joe mentioned, we're super grateful for everybody that has indicated that they would like to be a volunteer in one of these working groups through the RFI, and we're still working on forming those groups. So they're definitely not formed yet all the way. People that are asking in the chat, I know that people are very interested in knowing that, so still forming them. So the portal was opened on September 30th, and just for the data that I'm going to talk about today, the last poll that we did was earlier this week on November 18th. And so that equates to 49 days worth of submissions. And so totally we've had 313 submissions, and they were put in by 232 distinct individuals. Next slide. So when you look at the people that have been submitting to the portal, you can see that patients overwhelmingly have been the highest submitters. So 72% of the submissions we've received have been from people that have identified as patients of long COVID. And then the second most prevalent category has been individuals who are from a university. So that has been 11% of submitters, followed up by people that identify as caregivers, and that's 8% of the submitters. Then companies have been 5% of the submitters, followed by 4% of the submitters being from a research institute. Next slide. So when you look at the actual agents that have been coming in, like I said, we've had 313 total agents as of November 18th this week, and 69% of these agents have fallen under the category of drug. We've also devised a couple of other categories that we can kind of bucket agents into. So in addition to drugs, we also have nutrition and diet, which has been about 11% of submissions, manual and physical therapies, which accounts for 8% of submissions, devices, which has been 7% of submissions, and complementary and integrative health, which has been 5% of our submissions. Next slide. Because we've had so many submissions fall under the drug category, we've also subcategorized these submissions into different categories that'll just make it a little bit easier when we go through the agent review process. So the categories that we've subdivided into are immunomodulatory agents, which is about 45% of the drugs submitted, neurological agents, which is 24% of the submitted drugs, closely followed by antivirals, which has been 21% of drug submissions. Host-directed has been 8%. And then we have an other category, which is only 2%. This has been a couple of things that haven't really bucketed nicely into one of these different groups. Next slide, please. This is my last slide I'll show before we can jump into the Q&A. But we just wanted to highlight some of the specific submissions that have come in so far. So these are not agents that are being prioritized necessarily. Like I said, they still have to go through the formal scoring process. But we just wanted to highlight that individuals are really keyed into this topic, and people are having a lot of common ideas for what they're submitting. So the most common submission that we've seen so far is naltrexone, closely followed by COVID monoclonal antibodies. And then if you could advance one more, Joe. So then, like I said, kind of where we're at now. So we thank everybody again for filling out the volunteer form in the RFI. We've been collecting all the names of these volunteers. Soon we'll be sending out formal invites to populate these working groups. We'll then also have to send out some confidentiality agreements because some of the information in the portal may be proprietary. But once the CDAs are returned, then we'll be able to provide agent materials to these reviewers, and then that will begin the formal review process. So these agents will all be scored based on different criteria. And again, we're aiming for this first wave of agent review to be mid-December. So that's all that I have, and we can kind of transition into the Q&A now.
Thanks, Lizzie. This is just make a couple of comments. First of all, thank you for everybody. We have over 700 people participating, so that's an extraordinary rate of participation, and I think again speaks to the urgency that people really feel to figure out what's going on here, which we certainly understand. I think there is occasionally some frustration in the chat with the pace at which things are going. And I can tell you that part of that relates to just some of the logistics of setting up what essentially is a new parallel network to the old Recover mechanism. And that, of course, as you might imagine, it involves figuring out ways to get the resources that we need to do this work to our teams. So part of that is what we're working really hard on, and hopefully will inform the December initial December review. I will answer one question in the chat. Someone asked about composition of the working groups. We do want a wide range of people on these working groups. And someone asked about reimbursement. We are planning to reimburse people the same way we would reimburse people for participating in a study section, for those of you who know what those are. So it's not exactly a huge amount of money, but at least it is it's a recognition that these things take time and that your time is valuable, especially if you're struggling with some of these conditions. So Joe, I don't know, or Julie, if you want to add anything on top of that, just generally.
Julie?
Thank you. I don't have much to say other than that I too am reading the chat, and you know, we feel the urgency also. I kind of not being in the NIH, but looking upon the NIH, I can just tell you all of the things that are going on behind the scenes, that my colleagues really are doing the very best they can to expedite. Just this morning, we're meeting once or twice a week, every week, with the senior leaders. And this morning we had a quick review of the work group membership and how can we make sure that we are inclusive, but also optimizing the expertise and the lived experiences that go into this. So I think the December timeline to prioritize projects and get the work groups organized is ambitious, but we are doing everything we can to move that agenda forward. And we hear you. It sounds like a soundbite, but I mean it sincerely. We really do know how urgent this is.
And actually, Lizzie, I'm wondering if this is a good time for Julie to address the very first question that we thought was probably top of line and which has come across in the chat a lot. So would you like to go ahead and pose that?
Yeah, so we have a lot of individuals that are interested in knowing if we have an idea as to what the impact will be of the new administration coming into office later this year for long COVID funding and research.
No, of course, that's on everyone's mind, as it is every time we have a change in administration or a change in NIH leadership for that matter. These are very early days in that process, and there is a lot of opportunity for rumor and exaggeration and panic, I would say, in some circles. What we need to concentrate on is that long COVID is a public health priority, and the Congress recognizes that. We have bipartisan congressional support for this work, and we need to focus on it and prosecute our work as quickly and as expeditiously as possible. I would also say again, as much as this can cause anxiety, it's also a time to kind of step back and realize this, we are a very resilient organization. We've been here and gone through this before, and NIH will continue to do its good work. I don't personally, as a leader of an organization, spend much time speculating about what might happen or focusing on the what ifs. I have confidence from my long tenure in government and seeing many changes of administration that ultimately, when there's something important that needs to get done and NIH needs the congressional support to do it, it happens. And I don't think anybody's arguing that this isn't an important priority for our government, but also for our whole scientific community. So I don't want to sound like a Pollyanna, but I feel from a realistic point of view, this work will get done. It is prioritized, and even if there are significant changes in the administrative overview of the NIH, the work will go forward.
Great, thank you, Julie. And I just kind of want to build off that a little bit when we talked about funding some. There's some individuals who are interested in knowing if there's a ballpark number of how much money is left from the 1.15 billion that was appropriated for Recover in December of 2020.
So I can take that. So the majority of those funds are planned for use to finish out the activities that started in 2020. Some of them are already winding down, but you know, the adult cohorts recruitment and collection of samples is ongoing and is not yet finished. So while the money might not be officially spent, it's planned to be spent. So that's really the answer to that question. And that information is actually available on the Recover website, how that breaks down between the different Recover programs, whether it's observational, electronic health records, et cetera, and the administration of how to put it all together. You know, when Dr. Burton, I'll just remind people that when Dr. Bertinoli announced the $515 million appropriation and that RECOVER-TLC would occur, that was a different appropriation. So this program is coming from that 515, which has already been appropriated for this specific purpose. So I think we're working really hard to figure out the best way to do this, and we have those resources to do it. We just, you know, it's been just a few months since the meeting, and you saw what came in, and so we're really moving forward and recruiting experts. So I saw a few questions in the chat about, is, you know, this organization participating or is this one? And I would just encourage people to nominate, to put their name and their expertise and how they can contribute into the database, because while we all have colleagues that we know, it's better to have a pool of people who can broaden that pool, and that's really what we're asking for.
Great, thanks, Joe. And then, of course, we're talking about agents in the portal that we've seen submitted that will ultimately go into a future clinical trial under RECOVER-TLC. So we have some people that are interested in knowing if RECOVER-TLC plans to extend the clinical trial network utilized in RECOVER. There have been a lot of people that have experienced issues in getting to RECOVER sites if they live too far away from them. So there's just kind of some question on trial sites and if they'll be expanded upon in the current state.
So we heard that if you don't mind me, Jeannie, I'll answer.
Not at all. Go ahead, Joe.
Yeah, so we heard that message very clearly at the meeting in September, that we needed to think about how to broaden, you know, the hours that are available because people have to travel, and think about ways to do studies that could incorporate people who are really sick, who just simply could not travel to a site. So we aren't being constrained, at least intellectually at this point. And we would certainly like to leverage the expertise from RECOVER sites if it makes sense for the interventions that are chosen. And there probably will be some obvious overlaps, but there may not be complete overlaps based on, you know, what has come in the door, and, you know, to balance out what's occurring in the current RECOVER trials with things that really need to be tested based on what we know now.
That's great. The only thing I would add to that, thank you, Joe, is we have been in regular contact with the RECOVER investigators. So the original RECOVER investigators, we've had several calls. We want to make sure that this is as seamless as possible because, you know, they're still continuing to follow patients, right? The other thing I think it's important, just from an ethical perspective, you have some people who've been participating in RECOVER now for more than 2 years, who've been really dedicated participants, and we want to make sure they have the option of enrolling in some of these clinical trials. So there's a question about whether we can expand some of the sites beyond the RECOVER sites. We would like to take advantage of many other clinical trial sites that we have in our various networks. You know, we've got networks that are complementary to the RECOVER site team. So in a really great world, we will have the resources to offer whatever studies we're going to step up or stand up, not just to RECOVER people, or at least subsets of them, participants, but also to people who, like the person who asked this question, might be 2 hours from a RECOVER site, because that's not, again, we recognize that's not at all tenable for most people. But that's going to depend a lot, I think, on what we find out about the FY25 budget, which, you know, not to get too into the details, we're still under continuing resolution. I think some of you know that. It goes all the way to December 20th. There are discussions about what's going to happen with that. Will it be, will that end with the incoming new administration, or will it go into March or longer? We don't know. So that's definitely going to affect some of what we have the latitude to do.
And can I just say, and correct me if I'm overstating it, Jeannie, but you know, one of the things we learned from ACTIV is that you don't necessarily have every trial designed and starting at the same time. You know, this is likely to be a phased process, and we want to get the most promising things started as quickly as we can. But as the budget and the, you know, the science allows, these trials will evolve, and much better to do well planned and well executed trials than to race in and do a lot of small, statistically unhelpful trials that don't really give the answers that we need. So we're trying to get a balance here of getting it right, but also recognizing that we probably won't have every single trial plan when we come out of the working group process. We will be implementing and learning and adjusting as we go. And that, of course, will also depend on our ability to sustain congressional investment in the process, because there are limitations to how much we can do, even with the funding that has been procured already at this point in time. Am I saying that correctly, Jeannie?
Absolutely. And I think, one thing we did talk about at the workshop, and one thing that would be considered in a big way, I think, in the working groups is this concept of, do we look at a big clinical trial, say maybe 600 people in each arm or 390 people in each arm, so a pretty big one, to study something that we think has promise, or do we start out looking at smaller studies that can give us really solid information on mechanisms that might inform the choice of agents for bigger trials going forward? And I think, that gets to both the science prioritization as well as being good stewards with the money we have, and not knowing how much we're going to have, in the near future, hoping for the best, but planning for maybe not quite as much as we had hoped. But again, we really just don't know. So that's going to be a big focus of the working groups. It's also going to be a huge point of discussion for the overall scientific management committee, or I forget what we've actually called that, but sort of the committee that really integrates all the input from the working groups. So a lot to talk about there.
We've also had some questions about kind of the populations that we plan to include in future clinical trials under RECOVER-TLC. So specifically, people are curious to know if there are plans to include children, pregnant and lactating women, as well as individuals who have infection-associated chronic conditions and illnesses such as ME/CFS, even if they were diagnosed with that prior to long COVID.
So I think we heard that message very clearly at the meeting. I think particularly strongly for those communities you just mentioned. And I think we'd like to be able, it really will depend on the interventions and the prioritization. And if they can be tested in the pediatric population and lactating women population, we'd like to be able to do that. Of course, it's related to the resources, but we certainly would like to be able to broaden it, because to date, RECOVER certainly hasn't been able to address that. But it really is going to be a marriage of those opportunities and interventions, and then the safety and those other factors that need to be taken into account. But we recognize the need. And with regard to ME/CFS, I think it's quite clear, and there's been reporting from a number of the RECOVER cohorts that ME/CFS has developed as a result of COVID infection. Those, unfortunately, people who meet the criteria for ME/CFS after COVID infection. So I think they would clearly fit. And I think the question of adding folks who had ME/CFS before is one that question will be repeated across as we decide how to stratify, or whether we should stratify inclusion criteria based on the question being asked. Again, that was a really strong point from the community in the meeting, that we need to do that with our community input, because anytime you do that, maybe you refine the scientific question, but you might limit the people that might benefit or may need the benefit, in fact. So we're very cognizant of that, and we're open to trying to address as much as possible those populations. And I do think, because of the symptom overlap, it's a natural question, and we certainly hope that the benefits extend, but we really have to formulate, with the interventions and go at that direction, as opposed to design from the other way. But we are hearing that message, and again, working closely with our community members, we expect to work together in this program to continue to reflect, those asks.
And if I could just add on to just reading a couple of comments in the charts, one in the chat, one person said exactly what we've been talking about was that if you can't include people who are severely affected and even bedbound, the participant representation is going to be skewed, right? Because you're going to select for a bunch of people who are necessarily more, mobile and can still participate. So I think to really reflect the full spectrum of this disease, we really want to try to enable all kinds of participation, including from people who are bedbound. And now, depending on the intervention, that, as Joe said, that may be harder. But please know that we're thinking about that. A comment on including many of the other serious post-infectious chronic inflammatory conditions. This is a delicate line to walk because you know that this was an allocation specifically for long COVID. We recognize that there's a lot of overlap, probably with the mechanisms and with the symptom complex. We are absolutely committed to making sure that people are as represented as possible. There will be, though, a need to, if we're going to continue to keep this focused on long COVID, make sure that we do address some of the mechanisms that are specifically related to that virus. So I just throw that out there because I want people to realize that, we can think about a network of that focuses on people with chronic infection associated conditions, but we cannot be that network right now with this degree of investment. And I think that's a message people probably don't want to hear. But I think it's important to be honest about it because no matter how important all of those things are, we've really got to focus on at least one thing right now, do it well, and very hopefully those results will yield insights that can help us argue for the resources we need to study and move into some of those other populations.
I've also been seeing a lot of comments in the chat about the working groups, and so I just wanted to quickly provide a little bit of context around those too. So I just want to reiterate that these are really cross-sector groups. So we're working very diligently going through the list of all the volunteers that we've received, and you're welcome to keep filling that out. We're still accepting people and through February for the first groups, but we're really making sure that we have a good representation from all areas basically across the field. So industry, government, academia, people with lived experience, whether it's patients, caregivers, care partners, all across the board. So we're working hard to make sure that there are several of these individuals on each group so that it's not just one person that we're relying on to get, the impact from a certain community.
Yeah, I saw a question from, I think it was somebody from the VA. Is there a formal collaboration agreement? And I think we're hoping to pull in as many experts as we can who could contribute to this. So I would ask people to self-nominate if you have expertise that would fit this problem. And we already are pulling in those that we know about that make sense, but it really helps us if people tell us that they have an expertise from a different missionary. VA, has connections to a different population. We had Dr. Ziad Al-Ali at the workshop who, really was very helpful and offered his support with the things that have been learned taking care of patients in that network, as well as, the observational studies. So I think we don't have a formal collaboration, but we're certainly hoping that all those experts from those groups can participate to the greatest extent possible in the working groups.
We've also had some questions coming in, just asking if RECOVER-TLC is focused more specifically on launching clinical trials, or if there will be any kind of emphasis on providing grant funding to individuals to continue preclinical work as well.
So when Dr. Bertonoli talked about the launch of this, that came with the appropriation, she did mention that there were other activities that would come along. They're not quite the same as RECOVER-TLC. TLC is clinical trials. Could also and probably will include mechanistic clinical trials where we can get that kind of information. But to date, most of the pathobiology studies are coming from the broader RECOVER and not from TLC. And there, as well as the studies that occur across NIH that are still within the missions of each of the institutes that are supported in the traditional means. That does happen also. And we encourage that, and they're being submitted and supported all the time. I mean, of course we wish there were more, but that is happening. So there's several different ways that the pathobiology projects are occurring. And Jeannie, I don't know if you have any other thoughts there.
Just that one of the things we heard at the meeting was frustration about the lack of awards that had been made to the RECOVER investigators for pathobiology proposals that were submitted now almost 10 months ago or more under what was supposed to be a rapid process. We heard you. We really have helped, I think, to accelerate the review and handling of the proposals that were received, and we're very hopeful that you may be hearing something about those in the next couple of weeks. I think that's optimistic, but that's what we are aiming for and advocating for.
And then in terms of some of the agents that have come in through that portal, are there any plans to potentially combine agents into a combination therapy to test, or are you focused more on testing one agent at a time?
I mean, I think that depends on the study design. I definitely could see in the smaller mechanistic studies where you could test one and then modify based on what you learn, because that's a lot. Again, that's the feedback that Jeannie mentioned that we heard at the meeting, that the community wants that quick turn where you could really do that kind of combination therapy that wouldn't be so sort of onerous as a larger pivotal trial. That I think I could see that. But again, we need our working groups and experts to help us to sort through that, to get to the answer to that kind of question, to be honest.
Could just address something in the chat. Someone asked, we're using the term working group as if everybody knows exactly what that is. When we say working group, we really mean a small group of people with expertise in a given area. So for example, probably going to want a pulmonary working group, right? Because so many symptoms of long COVID are related to breathing. Probably going to want a neurologic working group because of all the issues, et cetera, et cetera. So those working groups will be set up to have expertise in specific areas that we really want to have good input on for these proposals. We also want people affected by those problems to be on the working group. So yes, scientific expertise is important, but again, what we've been hearing and what we have typically used for some of, I think, our best community engagement in infectious diseases is to have some representation from community patient in those working groups to be sure that we are not just having an isolated perspective from people who are reading the literature and doing scientific literature often, peer-reviewed scientific literature often, and basing their decisions on that. So I think that's an important point. And you can volunteer for the working groups. I believe there is we can provide the information for that to happen.
I'd like to just cover something on the submissions into the portal. There's been an ask as to what type of things will be taken into consideration. So we will be looking for things that are ready for a phase 2/3 trial. So again, just in the interest of, we hear you that we would like to get therapies out to be able to treat patients as soon as possible. We will be placing slightly higher priority on those agents that are ready to be tested for larger trials that could be then validated by the regulatory agencies. That doesn't mean other things won't be reviewed as they come along, but we just want to be very transparent that we are looking for things more ready to be out on the market.
And I do want to emphasize the regulatory agency. So if you weren't at the meeting in September, I encourage you to listen. It is available. I believe it's recorded online. Somebody asked about that. We can provide that link. But the last day we had really excellent discussion from the FDA, US FDA, and the European Medicines Association, which is the counterpart in Europe to the FDA. And it's really critical to know that we are in communication with them, and we will be in communication with them as we design these trials, because we want to really pay attention to what's going to get approved, because that relates then to reimbursement, insurance reimbursement, access, all kinds of things. It's not to say that something that is not yet FDA approved might be useful and might be studied, but I do think it's important to know that we are in very consistent communication with the regulators so that we can make sure we address any concerns or wishes as proactively as possible. In other words, we don't want to do a study, get a signal, and then find out we didn't ask the questions or get the endpoints that the FDA would have required to make a regulatory decision about approval. That's what I was trying to say.
And then another question that's come in a couple times is if researchers are submitting ideas to the portal and say their idea gets suggested as something that could go forward for a clinical trial, will those individuals be involved in any of the planning or implementation in regards to the trial?
I mean, I think honestly, that was one of the challenges with the way that RECOVER took in ideas and then they were disassociated from the investigator who proposed them. So I think we wouldn't want to repeat that. I don't think that was productive. I mean, I think it will, like any protocol development, it will take many points of input, but I don't think we'd want to eliminate the person that suggested the idea.
So we'll have time for a little bit more questions, but I just kind of wanted to open it up to everybody on this to see if there was something else that Jeannie or Julie or Joe wanted to make sure we touched on before we get close to having to wrap up.
A really, really excellent question about somebody saying, I don't understand why the emphasis on phase 2, 3 trials when there's no pipeline. Wouldn't it be more wise to do a phase 1, phase 2? That is exactly the question that we need to wrestle with. And what I was mentioning before, and what Dr. Gerberding mentioned too, about how you figure out what the most important questions to ask, given the spectrum of agents you have right now to do a study with, right? Like, are we really ready to do a phase 3 trial? Some people may argue that we are, and that's where we would have the debate when the proposals are submitted. Other people will say, we're not ready, as this person in the chat said. We may need to do some smaller phase 2 trials where you enroll a couple hundred patients or 100 patients in each arm, and you really look mechanistically with immune signatures, also get detailed assessment of whether people are feeling better, just as signals of whether to move forward. And that's where we talk about this platform design, where you don't put all your money immediately into a giant study when you don't exactly know for sure whether it has a really good chance of working. So that's going to be part of the really hard work of this group. And I think one of the reasons it's exciting to have it coordinated through a new network.
I see some comments too about establishing clean endpoints, and I just want to reiterate, I think we've kind of touched on this, but we are actively working with the FDA to try to align on those things, and it is at the forefront of our minds as to how important that will be when we end up coming to design these clinical trials.
Yeah.
And +++1 for biomarkers. Be great to have something that we could use to complement our functional measures, right? Functional being how you feel, I think that has been tough so far, right? We've had a bunch of, even RECOVER looked pretty hard at this and was not a really positive yield. So I do think, again, that's part of something we would build into some of these trials. And I agree, asking people how they feel is really critical. I don't know that, I mean, whether you call that a biomarker or not, when we think of biomarkers, we usually think of molecular mechanisms that signal a process that you have changed by giving the drug. But they're both hugely important, and we need to figure that out. And one of the things that we might find from mechanistic smaller trials is, again, hunting down these biomarkers.
I noticed there's been a lot of chat about repurposed drugs, and I think that's that was reflected in the list that Lizzie showed, actually,
Mm-hmm.
which will probably more likely meet some of the criteria that Stacy mentioned, safety profiles and availability, obviously. So those are all strengths, and so we're noting that.
And I'd also note that, we can't really talk about what the pathogenesis awards are going to be, but there's clearly people who are interested in pathogenesis are interested in biomarkers. So getting some money out the door for those studies finally may advance that front, and that's going to be really, really critical.
I think we're at 3:01.
Yeah, we're right at time. I just want to remind everybody that this is being recorded and we will be able to post a recording so you can watch it back. We're also capturing the chat, so of course we weren't able to get to everybody's questions, but in addition to the webinars that we're planning to do, like Jeannie said, there'll be another one in January, but we're also planning to supplement those with newsletters. And so perhaps in the next newsletter, we'll try to address some of the questions that we weren't able to get to today. And that should be in mid-December when that will be going out.
Well, if that's at time, I really want to thank everybody for their incredible engagement. Again, we've managed to keep almost 500 people on the call, and I know there's a really urgent need for more specifics. We don't have a lot of them, but we are definitely keyed into the need to come back to you in January with hopefully a lot of work that we'll manage to do in the next couple of months, and some more clarity about what's going to happen in 2025, more importantly, or equally important. So, and thanks to our team at FNIH and all the folks at NIAID who worked very hard to put this together. So really appreciate y'all. All right. Thanks, everybody. Have a good day.
Bye-bye.